Welcome to this tutorial on LFPy. My name is Torbjørn, and I am one of the main developers of LFPy, which is a Python tool to calculate electromagnetic brain signals from simulated neural activity. Before I go into the details of how to use LFPy, I will start with a very brief motivation and give some background information. In neuroscience, we often try to understand and make claims about neural activity based on recorded electromagnetic brain signals, like extracellular action potentials, local field potentials, EEG signals, or MEG signals. However, linking recorded brain signals to neural activity can sometimes be difficult, and there are some pitfalls. At our group at the Norwegian University of Life Sciences, we have been working on the link between neural activity and the resulting brain signals for about 20 years. Through this work, we have learned that instead of relying on our intuition and trying to reason about what different types of neural activity will look like in a given brain signal, it's often best to just run numerical simulations where we simulate different types of neural activity and calculate the resulting brain signal. And going the other way around, when starting with some observed feature in a recorded brain signal, we have learned that simulations are often very helpful to help us understand what this feature actually represents in terms of the underlying neural activity. To do these kinds of simulations, we have developed the open source Python package LFPy. The name LFPy reflects that the original purpose of the software was to simulate local field potentials. But LFPy can also simulate extracellular action potentials, ECOG signals, EEG signals, and MEG signals. Here are a few example plots of different brain signals simulated using LFPy. This is an example of simulated extracellular action potentials, or spikes around a detailed cortical pyramidal neuron created by Hey et al. In some cases, for example, in vitro slice recordings, the extracellular environment of the neuron is highly inhomogeneous, and this can be taken into account by LFPy. LFPy can be used to run single-cell simulations, but also large-scale network simulations. We can also use LFPy to simulate EEG signals where the complex conductivity and geometry of the head must be taken into account. Many more examples can be found by going to LFPy's GitHub page, to the documentation, or by searching for LFPy on eBrains. Originally, LFPy was built as a Python interface to the neuron simulation environment, and it was used both to set up and control the neural simulations and to calculate the resulting brain signals. However, LFPy has now essentially evolved into a family of different but related software. At the lowest level, we have LFPy Kit. LFPy Kit is a freestanding implementation of all the so-called forward models incorporated into LFPy. A forward model simply means that it takes you forward from the simulated neural activity and to the resulting brain signal. LFPy Kit is simulator independent and cannot in itself be used to run simulations of neural activity, only to calculate the brain signals resulting from neural activity simulated elsewhere. LFPy Kit is not tied to the neuron simulator and can be used with, for example, Arbor, but also with other neuron-based software. Next up is LFPy. LFPy depends on LFPy Kit for calculating brain signals and can be used as a convenient Python interface to Neuron to set up and control neural simulations. This is in many ways the main software, and it is the software that I will soon demonstrate how to use. Before I go on to demonstrate LFPy, I will briefly mention two more advanced members of the LFPy family, namely Hybrid LFPy and LFPy kernels. Note that these two software packages are intended for more advanced users, and I will not go into much detail. The motivation for making Hybrid LFPy and LFPy kernels was that many brain signals, like LFP and EEG signals, represent neural activity at the population level. And simulations of neural activity at the population level are often done using simplified schemes like large-scale point neuron network simulations, for example, in NEST, or through neural mass or neural field models, for example, using the virtual brain, or TVB. At the fundamental level, electromagnetic brain signals are caused by the spatial distribution of memory currents over the individual neural morphologies. And since simulations of neural activity using simplified schemes often do not explicitly model the spatial structure of the neurons, they are not in any principled way able to predict extracellular potentials or other brain signals. Hybrid LFPy and LFPy kernels represent different tricks to overcome this problem. Hybrid LFPy is intended for running large-scale simulation with point neurons, typically in nest, while still calculating brain signals from the simulations. Essentially, this is done by first simulating the neural network activity using point neurons, and then afterwards, in a separate step, replay this spiking activity as synaptic input to representative morphologies representing the different populations. From this, we can then calculate the brain signals. For more information on this, I recommend Hagen et al. 2016. LFPy kernels is intended for calculating brain signals directly from simulated firing rates. The concept is a bit challenging to explain briefly, but the core idea is that for each connection pathway between a presynaptic and a postsynaptic population, we can estimate the average contribution to a given brain signal from the postsynaptic population stemming from one action potential in the presynaptic population. This gives us the so-called kernel from this connection pathway, and by convolving this kernel with the presynaptic firing rate, we obtain the approximate contribution to the given brain signal from the considered synaptic pathway. By summing the contribution from all the synaptic pathways between the different neural populations, we get the simulated brain signal. For more information on this, I recommend Hagen et al. 2022. As I said, hybrid LFPy and LFPy kernels are intended for more advanced users, and I will now return to the main software LFPy and show a simple example of how it can be used. For this demonstration, I am using the eBrains Collaboratory. I'm here assuming that you are familiar with basic Python programming and with how to use Jupyter notebooks. We first tell the notebook to display the plots, and we do the necessary imports. Conveniently, LFPy is pre-installed on the eBrains Collaboratory. Now we make the cell class. The only required argument here is the morphology. Morphologies can be obtained from many different repositories, for example, neuromorpho.org, ModelDB, or through the eBrains knowledge graph. Here we use the Meinen-Szynovsky cell model morphology that comes with LFPy. To keep it simple, we also use the passive equals true argument to make LFPy insert some passive parameters to the cell model. For plotting purposes, we also rotate the cell so that it is aligned with the z-axis, and here I just do this manually. Now that we have a cell model, we need to give it some input. There are tools in LFPy which can be used to find synapse locations on the cell, but here I will just use the compartment index 0, which typically means the soma. Then we have the strength or the weight of the synapse, the synaptic reversal potential, and the synaptic rise and decay time. I also tell the simulator to record the synaptic current for plotting purposes. Now let us set the synaptic activation times, and here we tell the synapse to be activated at 30, 35, and 40 milliseconds. Now we're ready to run the simulation. And we want to record the memory currents, as this is the basis for calculating extracellular potentials. Let's then add an extracellular electrode to record the extracellular potential resulting from this single synaptic input. We set the extracellular conductivity to 0.3, which is typical value, 0.3 Siemens per meter. And we make a single electrode, 20 micrometers from the origin in the positive x direction, that is quite close to the soma. Then we make the electrode with LFPy's rec_ext electrode class that is recording extracellular electrode. Now, to get extracellular potential from the membrane currents is essentially just a matrix multiplication, which we can write like this. We have now calculated the extracellular potential from the simulation, and we can plot the results. Here, we plot the cell morphology in black. We plot the synapse location with a red dot. We plot the electrode location with a blue dot. We plot the synaptic current, the somatic membrane potential, and the extracellular potential at this electrode. So this is a very simple example of how to use LFPy. There are, of course, many more examples available. For example, we can look at the file name LFPy example 0.4. Here is an example of spike waveforms from the Hay et al. 2011 cell model. This might look more complex, but it's mostly because the cell model is downloaded and compiled, and also because the plotting is more sophisticated. But here is the result of how the different spikes can look like around the Hay model neuron. We can also look at LFPy's example 08. This is a passive cell model from Meinen and Chernowski, and it is receiving 3 different kinds of synaptic input with more complex spatial and temporal properties. And here is the result. Of course, there are many more examples available, including neural networks, EEG signals calculation, and stimulation by extracellular current injection. So if you're interested in finding out more, I suggest that you have a look at the examples that comes with LFPy. Lastly, I would like to take this opportunity to mention that we have written a comprehensive textbook on electric brain signals with a special focus on how to model them. The book contains chapters on neural dynamics, volume conductor theory, spikes, LFP, EEG, ECOG, MEG, and more. And it has dozens of code examples using LFPy. So, of course, if you have any more questions, feel free to email us or to open an issue at GitHub. And thank you for watching.